Good evening, sir.
Yeah, good evening. We are just waiting for a few more to join and then we will start.
Okay, sir.
Okay. So, yeah, good evening everyone. Those who are new and might not know me, I am Srijan Sen from IIT Kharagpur, and I will be taking the live sessions on this particular course. Okay. So we will, like I do, so we will be first going with the lecture slides of week 2 in brief, and then we will do some question and answers. Okay. So, right. So let me share my screen with you all. So I guess my screen is visible to you all. Okay, cool. So in today's class we will be, so last class we talked about nucleic acids as a genetic material, and then we discussed about the DNA replication, and we started with this replication part. This class our main topic would be to understand the transcription and the translation, that is how the information is produced and stored in the DNA. Then it is made into some RNA and then finally translated into protein. So starting with where we stopped last class, and we were discussing about replication. So we understand what DNA replication is now, and what are the leading players. The important players over here is the leading strand of you have the leading strand of DNA, then lagging strand of DNA, you have the DNA polymerase. Okay, you have the DNA polymerase 3 helicase, primases. You have the RNA primers which are being produced by the primase, the Okazaki fragments, and the ligase. So these are the important players, the major proteins which play, or the major segments that play an important role in replication. So some important, just a recap of some important proteins and enzymes that were playing a role. So we have the DNA helicase that unwinds the double helix at the area where we have the replication fork, or where there are stringent folds. So unwinding of those helix regions is important. Now as the two DNA strands separate and the bases are exposed, then comes the role of the DNA polymerase that moves to a position from where the synthesis begins. And how does this synthesis start, or the production start? For that you need the help of RNA primers. Why RNA primers? Because these are single stranded small segments, so primase plays an important role in production of these RNA primers. Now you have the leading strand which is synthesized continuously, but the lagging strand is synthesized discontinuously. We discussed last class what was the reason behind that. So after the DNA is synthesized, you have another DNA polymerase that removes the RNA primers and replaces them with the DNA. Okay, so if you are wondering about the first answer to the first question, that is why is the leading strand synthesized continuously but the lagging strand is not, then you might be aware that the DNA polymerase can only synthesize along one particular direction, that is the 5' to the 3' direction. So you understand, taking the complementary strand of the 3' to the 5' strand, which is originally present in the template region, you have the DNA polymerase that will extend from the 5' to the 3' end, because it has the 3' to the 5' end. Taking the help of the 3' to the 5' end, the DNA polymerase continuously elongates across in the 5' to the 3' direction. That is why it's the leading strand. Whereas for the lagging strand, what happens, it's the 5', it has to take the help of the 5' to the 3' strand, but it cannot synthesize in the 3' to 5' direction. So it has to generate small fragments of, or pieces of DNA across the 5' to the 3' direction, and that is known as the Okazaki fragments. So you see over here, this is the template, the parent DNA, it unwinds, and then the leading strand is produced, which is the 5' to the 3' direction, and then you have another lagging strand where it is produced in the reverse direction. So it starts from the 5' to 3' again from this side and this side, and then it's joined by the ligase enzyme, right? Okay, now our main topic of interest in today's class is basically transcription and translation. So we need to understand what is transcription. So it is the process of making copies of genetic information that is already stored in the DNA strand into a complementary strand of RNA. That can be the messenger RNA. Why is it a messenger RNA? Because it carries the message that was previously stored in the DNA and is transmitted to the RNA. So it's a messenger RNA, and it is done with the help of an RNA polymerase. Like in replication, there are roles of DNA polymerase. So for transcription, as it's from a DNA to RNA, it's a conversion from a DNA to an RNA. That is why comes the role of a RNA polymerase. So in short, transcription is the process by which the RNA molecules are initiated from the DNA. They are elongated, terminated. That is called transcription. So from here you can understand that transcription, like replication, has multiple steps. Transcription has also three major steps that they are divided into three major steps, starting from initiation, elongation, and finally termination. So transcription site in prokaryotes and eukaryotes. So eukaryotic transcription takes place in the nucleus. So you have a DNA, then the RNA polymerase comes and a pre-RNA is produced, that is transcription. Then you have the mRNA, which is put up into the ribosome for the translation. And prokaryotic transcription takes place inside the cell, as there is no separation by the nuclear membrane. So the location of the transcription is very, very important. Now there are basic features of RNA synthesis. So precursors, that's the 4 ribonucleotides. You know that over here when it's an RNA, there is no thymine, which is substituted with an uracil. So precursors, the 4 ribonucleotides, the 5' phosphates, ATP, GTP, CTP, and the UTP are required for the synthesis of RNA from the DNA. Now there is a polymerization reaction that takes place. It happens in a similar manner as it's for a single-stranded DNA, where a 3' OH group of one nucleotide reacts with the 5' triphosphate of a second nucleotide, and it elongates across one particular direction. So you must be wondering what would be the what would be the sequence of the RNA be, considering that there are 2 strands of DNA where it has a 3' to the 5', and another one is running across the 5' to the 3'. But RNAs are mostly single-stranded, generally single-stranded. mRNA is single-stranded, and that is the way the message is transmitted from a DNA to an RNA. So which strand will it take the help of? Let's see. So you see over here, the sequence of the bases in the RNA molecule is determined by the base sequence of the DNA, and there are 2 strands of DNA which we call as the template strand and the DNA coding strand. So DNA coding strand is the one that is the 5' to the 3' end, and the template strand is the 3' to the 5' end. Why is it a template strand? Because it plays a template. It is the starting material for the production of RNA. Okay, so that is why we call it as the template strand. Fine. Okay, so if that's fine, so taking the help of the template strand, which is originally the complementary strand of a DNA or the DNA coding strand, you can immediately understand that the primary sequence or the DNA coding strand sequence is same as that of a mRNA. Only difference that you will be seeing is basically all the thymines to be replaced by an uracil. So the DNA coding strand is exactly same as that of an mRNA or the strand which is produced. Just a minute. Okay, so I guess you understand that the template strand is the strand using which the RNA polymerase will produce the mRNA. Fine. Okay. Now the DNA molecule being transcribed is double-stranded, yet in a particular region only one strand serves as the template, because the complementary strand will be basically used for the generation of the mRNA. Fine. Okay. Next up, the chain growth. As I mentioned, it is similar to that what is being produced in for DNA. So nucleotides are added only to the 3' OH group of a growing chain. And over here you do not require a primer. So most of you must be aware that why it does not require a primer, because it already has a sequence, a template strand or a template sequence, which is a known sequence, and therefore it is important, it is unnecessary to have another kind of a sequence or bring in another primer for the generation of it. So the first base is to be laid down in the initiation event is a triphosphate. It's the 3' OH group is the point of attachment of the subsequent nucleotide. Thus the 5' end of a growing RNA molecule terminates with a triphosphate, and that is how the elongation proceeds. So what are the requirements for transcription, or what do you require for transcription? And that is basically the template, which is the double-stranded DNA, activated precursors, or the basically the DNTPs, you can tell, the ATP, GTP, UTP and CTP, some the divalent metal ions. You have the manganese, magnesium, things like that, and the RNA polymerase, which is the enzyme. And these four basically are good enough to carry out the transcription process. So what is the RNA polymerase? It's a key enzyme which was isolated from E. coli by Chamberlain and Berg in the year of 1962. If you can see over here, there is a core enzyme and an hollow enzyme over here. The core enzyme has 2 alpha units and 2 alpha subunits and 2 beta subunits, which forms the core enzyme. It's core enzyme to describe the alpha free unit, namely A2, beta and beta dash. You need not remember all of these in much details, but sometimes there are questions based on this. And there is another factor, which is the sigma factor. So this factor is basically the triggering factor, or sometimes you call them as the cofactors. So sigma factor is the one that activates the enzyme or completes the enzyme. So the complete enzyme, where you have the alpha 2, beta, beta dash and the sigma factor, it's known as the hollow enzyme. And this entire 5 subunits comprise make up the RNA polymerase. Fine. So yeah, sometimes these are important because you need these functions of these subunits are important because it's asked in a lot of competitive exams and things like that. So yeah, you need to know about them. So as I was saying, so you have the beta. Let's start from the beta dash subunit, which is of a molecular weight of 160 kilodaltons, and its function is binding of RNA polymerase to the DNA, that is to the template strand. The beta subunit, which is around 150 kilodaltons, and it helps in binding of the incoming nucleotides and forms the first phosphodiester bond formation. The 2 alpha subunits, which comprise entirely of the 40 kilodaltons, is needed for the assembly of the core enzyme and the recognition of the promoter site. And your sigma subunit is around 90 kilodaltons, and it helps in the recognition of the start signal and directs to bind to the promoter region of DNA. Therefore, all these 5 subunits, 5, or you can tell 4 if you're considering the 2 alpha subunits to be 1, then these 5 subunits totally combine up to play a very important roles in the RNA polymerase and in the transcription process. So there was a term promoter. So what is a promoter? So promoter regions or promoter sites, promoter sequences are sites in DNA molecule at which the RNA polymerase binds, and that is the starting site. Promoter site is the starting site where the RNA polymerase binds, and RNA polymerase transcription factors binds to initiate the transcription process of the mRNA. like you have the ORIC site, the origin of replication site. So here you have the promoter site from where the initiation of transcription starts. Now, as I was telling, transcription can be categorized into three major processes. That starts with the initiation, elongation, and the termination. So template binding at a polymerase recognition site, then binding to the initiation site that will lead to the elongation, and finally establishment of the open promoter complex with final termination to end the transcription process. Okay, so yeah, so over here we have a DNA sequence. You have the promoter site, and then after a stretch or a break, you have the coding sequence of genes. Then strong E. coli promoters over here. So there are certain sites that are identified by the RNA polymerase, like the TT. Then you can have something over here, and ACA. So you have a TAT and AT. So these are the different, so you call it the Tata box mostly. If you can remember about it, it's the Tata box over here. And then these are the certain E. coli promoter sites that are present. So consensus sequence of most E. coli promoters have around they are present at a stretch of like 15 to 17 base pairs. Fine. Okay. Now we will get into the initiation of the synthesis. So there are upstream promoter elements. You have the Tata box, and then after if you're considering from minus 35, so after 35 base pairs, or after 10 base pairs, if you're starting from the second site, or after 10 base pairs starts the mRNA initiation process. So RNA polymerase is a huge enzyme. It binds around 60 nucleotides at a time. So RNA polymerase, what it does, it scans the double helix. It goes and binds to the promoter region of the DNA. So the polymerase binds to the promoter region, forming a closed complex. So you can see over here you have a closed complex. It binds over here, and then the downstream signaling or the downstream initiation process begins. Okay, so you can see how it sits over here. So that is why this stretch is important. You cannot have the two recognition sequence very close by, because it requires a distance of 15 to 17 base pairs for sitting down. Right, then the polymerase unwinds the DNA, forming an open complex, after which you have the DNA coding strand, which is not being used. It uses the template strand. Okay, it uses the template strand for the production of the RNA. Yes, for the production of the mRNA. And then the synthesis continues. So 3' to 5', you see that it's continuing, it goes, and the process goes on. Now there is elongation of the chain. So it elongates. So this is the mechanism. You have the RNA polymerase, and then you have the -1th base, and then it binds to the 3' OH. The 5' phosphate goes and binds over here. The nucleotides are attached one after the other. So that's the that's the elongation process. So the RNA polymerase goes ahead and it starts adding the nucleotides one after the other by watching over the template strand. And finally, there is the termination of the synthesis. So termination of the RNA synthesis occurs at specific base sequences within the DNA molecule. These sequences are of two types: simple terminators and termination factors dependent. So what is simple termination? And the features are that the sequence in one DNA strand would read like ABCDXYZ, something like that. Thus the sequence is capable of intra-strand base pairing, forming a stem and loop in an RNA transcript. So you see over here, it's like this strand recognizes this strand, but in a complementary way. So it will form a loop-like region, and it folds, and the process ends over there. So that's simple termination. And the other one is factor dependent. termination factors dependent, that's the row dependent termination. So as the polymerization proceeds and cut sites are exposed, row factors recognizes and attaches to it. Now as the polymerization proceeds and C-reach segments are made, the ATPase activity of the row increases until further polymerization is processed. Now as no polymerization occurs, the RNA polymerase stops its action. However, since the no sigma factor in the core enzyme, it cannot restart the process. Hence there is separation of all the units and the mRNA gets released. Right, so what you see over here, you have this RNA polymerase, you have the sigma factor, so it goes and sits in the promoter region, it identifies, it opens up the DNA and it starts producing the mRNA. After that, the mRNA, you see there is elongation, there is preparation of the mRNA, after which what happens, there is some sequences within the RNA that either forms a loop or it's a row dependent termination, and finally the mRNA is released. Okay, so yeah, any questions up to here? If you have any questions, you can ask me. You can put it up in the chat box, anything that's comfortable with you all. Okay, so no questions? Fine. Then we will get into translation. Great. So getting into translation, that is, so by definition, translation is a process where the proteins are made by the ribosomes on the mRNA strand. So taking the help of the mRNA strand, ribosomes make proteins. So the process in the ribosomes of a cell by which a strand of messenger RNA directs the assembly of a sequence of amino acids to make a protein. So we had DNA, it was transcribed to an RNA and translated to a protein. That's the central dogma. Now, site of protein synthesis in eukaryotes and prokaryotes, you can see over here that this is the position, this is the location where the translation is taking place. So from the nucleus, it goes, the mRNA is released into the cytoplasm, and over here the translation is taking place for an eukaryotic cell. For a prokaryotic cell, it does not have the nuclear membrane to do anything. Right, so the ribosome, you see over here, it has there is a 70S ribosome, then there is a 30S, 50S. So these are the different ribosomes over here. So the different steps in translation is the activation of amino acids or the initiation of synthesis, then transfer of amino acids to the RNA, that is the charging of the tRNA, and elongation of polypeptide chain, final termination of synthesis, finally termination of the synthesis. So the role of RNAs in translation, so this is, you can see over here, this is a tRNA molecule, and it has to be charged with a particular amino acids. That's the charging of the DNA. So mRNA carries the genetic information, and proteins or amino acids are encoded in sets of 3 known as triplets. So it uses a stretch of 3 nucleotides to represent one particular amino acids. So rRNA, or the ribosomal RNA, is basically the integral part of the ribosome. Over here, what matters is the tRNA that transports that gets first charged with an amino acids, and then it transports the amino acids. So fine. So activation of the amino acids and charging of the tRNA, which is the most important part over here. So this is an amino acid tRNA synthetase. Then you have the amino acid and the energy source, which is ATP. So it is using this first. You have to charge the tRNA with that particular amino acid, okay, and then what you see over here, it is basically, so it's basically charged. Then the tRNA you see over here, so these are all the charging and the different arrangements that are happening over here. So these are the steps which you can remember, because what is important is the charging, where you have the amino acid tRNA, you have the energy source of ATP, you charge it with a particular amino acid. So every triplets or every codon will, every sets of 3 nucleotides will be responsible for the identification of that particular tRNA, and the tRNA is charged with an amino acid that will be transferred. So initiation of translation starts with an initiation factor 1 and initiation factor 3. So after they come and sits in the 3S ribosome, you have the initiation factor 2 that comes and forms the pre-initiation complex in the 30S ribosome subunit. So initiation factor 3 mediates or identifies the signed Delgado sequence in the 60S RNA, and from there the protein translation starts. So there is a site where the ribosome goes and binds. Okay, so ribosome identifies first the signed Delgado sequence. It goes and binds in the leader sequence where the ribosome goes and binds, and then you have heard of the start codon, that's the AUG, but the initiation factor 3 mediates the protein translation machinery. So it will start from the AUG. Wherever there is an AUG, the initiation process of translation starts and ends at a UAG. So there are 3 stop codons and 1 start codon. You have the amber, ochre and opal stop codons, and for the start codon you have only AUG. So previously there is a leader sequence where you have the ribosome binding site. This is the coding sequence. The coding sequence is basically the portion from where the protein translation will basically start. It will start preparing the amino acids because it does not go into the leader or the trailer sequence. And you must remember that stop codons do not have any tRNAs. So there are no tRNAs for the stop codons. Fine. Okay. So yeah, there comes another charging and the importance of the different initiation factors. So you have so that's how the initiation of the translation starts. Then comes the elongation, where you have the extension the elongation factors, the EFTU, the FTS and the EFG. So there are different factors, the peptidyl transferase center, active center, that are created by the L proteins. And then you have the A site, the what are the different sites? You have the A site, the P site and the E site. So E site is the exit site, P site is where you have so you have the A site, you have the P site where it starts the elongation, and from the exit it basically goes out. So peptidyl transference center or active center, so you have the formation of the L proteins or large subunit center. So you have the FMET, it is the starting one, and then you have the amino acid with the tRNA, it goes into the P site, and there is an uncharged tRNA. Okay, and in this particular A site you see you have the AUG, which is coding for methionine, and then from the P site there is elongation that is starting. So it goes over here. So as it goes, it keeps on adding the amino acid and it gets extended. When it gets extended, what happens? It goes into the P site. So then over here again it comes and this histidine is attached. So this happens across the entire mRNA sequence. And finally you would see that it keeps on adding. It keeps on adding until it reaches the stop codon. Stop codon is an indication that the stop codon will be an indication that there are no tRNAs for this particular triplet codon, and then your translation machinery stops and there is termination. Okay, so if we go like in a cycle, what happens? You have the ribosomes, there are the 3 initiation factors that starts the initiation process. You would form the pre-initiation complex. Okay, and then there comes the mRNA. It first identifies which mRNA it is going to code for. Then the tRNA has to be charged. The amino acid tRNA has to be charged with the particular amino acid, and then it reads through the mRNA strand, and then for every particular triplet codon there will be one particular tRNA that it has to find out, and it goes and sits. Okay, so there are no amino acids that are identified for stop codon because there are no tRNAs for it, and there is also no amino acids that will be prepared once there is termination. Okay, so there is an A site, P site and an A site. It enters through the A site. In the P site there is an elongation. It is again pushed into the P site. So every amino acid goes through this. In the A site it is identified, in the P site it elongates, and through the E site of the ribosome it exits as an entire primary sequence of the protein. So it starts with a methionine. We all know that is an AUG, and then there is elongation across while it reads through the mRNA sequence. Okay. Any questions? Two minutes if you have any questions. No questions? Okay, then we should scan through the quiz. Okay, so this is more of a what you people would be answering, right? So you can answer me in the chat box, and then wherever there are doubts, we can give you a clearance. Okay, let's start with the first one then. So which enzyme is primarily responsible for unwinding DNA at the replication fork? All of you should try to answer because these are not very difficult. So DNA helicase, because it's the enzyme that unwinds the double-stranded DNA helix at the replication fork, separates the two strands so that they can be used as a template for the DNA synthesis, new DNA synthesis. Okay? Yes, option B. Why are RNA primers required during DNA replication? Why do we need RNA primers during DNA replication? Yeah, we have so many people. Why I need answers from all. Everybody should try to participate. The questions are not very difficult. There is not feeling shy about it. If it's wrong, it's fine. It's an interactive class, it's a live session, so you people should be more interactive in that sense. Yeah. So DNA polymerase can only add new nucleotides to an existing strand of a DNA or RNA. It requires a free 3' OH group to start its or initiate its synthesis, and the primase is required for the production of the RNA primers, short RNA primers, providing this necessary 3' free OH group, allowing for the DNA polymerase to start working. So therefore option B, which is correct. The third one says lagging strand synthesis is discontinuous because. Why is the lagging strand synthesis in DNA replication discontinuous? Yes, DNA strands are anti-parallel. That is basically the reason. So the two strands of DNA run anti-parallel, and DNA polymerase can only synthesize in the 5' to 3' direction. As the replication fork opens, one strand, which is the leading strand, can be synthesized continuously, whereas the other one, which is in the 5' to the 3' direction, for the synthesis of the 3' to the 5' direction, DNA polymerase has to do it, is compelled to do it in the 5' to the 3' direction, and therefore which it cannot compromise on. Therefore shorter fragments are produced at a time, and that is why the process is slow as well as discontinuous. So yeah, option B. Which enzyme replaces RNA primers with DNA in prokaryotes? Yes, DNA polymerase, it is responsible for the removal of the RNA primers using its 5' to 3' exonuclease activity. It not only removes the RNA primers but also fills the resulting gaps with the DNA nucleotides. So option A. Let's go into the next set. Which bond is sealed by DNA ligase during replication? Hydrogen bond, glycosidic bond, phosphodiester, and peptide bond. I see only one person answering. This is not good. Everyone should try to answer. So which bond is sealed by DNA ligase during replication? So the phosphodiester bond, yes, that is correct. So DNA ligase seals the nicks in the DNA backbone during replication, repair, and recombination. Now these nicks involve joining of the 3' hydroxyl end of one nucleotide to the 5' phosphate end of another, which forms a phosphodiester bond. Now hydrogen bonds links the complementary bases between the two strands of DNA double helix and are broken by helicase during replication. Glycosidic bonds link the nitrogenous base to the sugar molecule in the nucleotide, and peptide bonds link amino acids to other proteins. So definitely it's going to be the phosphodiester bond. a very direct question. Transcription in eukaryotes occurs in the cytoplasm, nucleus, ribosome. Transcription in eukaryotes. Yeah, so in eukaryotes, the DNA is present in the nucleus, where the transcription, that is the DNA to RNA synthesis, takes place, and the resulting mRNA is then takes an exit from the nucleus to the cytoplasm for translation or proteins at the ribosomes. So option C, which is correct. Now, which RNA polymerase subunit is responsible for promoter recognition in prokaryotes? Which subunit is responsible for promoter recognition? It's the sigma factor, right? So in prokaryotes, the main or the core RNA polymerase enzyme requires the sigma factor, which is basically more or less like a cofactor to identify, recognize the promoter sequences. So its binding is in the -35 and the -10. With the stretch of 15 to 17 base pairs in between, and it sits over there and starts the transcription. So option number C, 7C, is correct. Question 8, the complete RNA polymerase enzyme including the sigma factor is called core enzyme, holoenzyme, apoenzyme, or the transcriptase? Yes, it's the holoenzyme, the whole enzyme. The core enzyme has only 2 alpha and beta dash. With the sigma, it's known as the holoenzyme. Okay, now question number 9. It says RNA synthesis. Okay. RNA synthesis proceeds in which direction? RNA synthesis proceeds in which direction? I've got one answer. So RNA synthesis takes the help of the template strand of DNA. The template strand of DNA runs 3' to 5'. So taking the help of it, the RNA polymerase sits in the promoter region and starts the synthesis across the 5' to the 3' direction. So option B. The next one says, which of the following is not required for transcription? DNA template, RNA primer, RNA polymerase, ribonuclease triphosphates. B, RNA primer, yes. So transcription requires a DNA template, RNA polymerase, and the NTPs. as the building blocks. So unlike DNA replication, transcription does not require an RNA primer for the synthesis, for the initiation of the synthesis. Now question 11 says intrinsic termination involves formation of. So intrinsic, that is row independent termination. So there is row independent termination involves the formation of protein complex, stem loop structure, ATPase activity, DNA nick. Yeah, so intrinsic or row independent termination means there has to be some complementary site that will come and form a loop-like structure. That's the stem loop structure. So it's more or less like an hairpin model in the newly synthesized RNA molecule, and then after the loop is formed, it will terminate. So yeah, stem loop structure, which is option B. Now, row-dependent termination requires alpha factor, ATP hydrolysis, DNA ligase, or a tRNA. Yeah, so yes, ATP hydrolysis, so row dependent termination requires some kind of an energy source for the action of the row protein, which is in the form of an ATP. So row proteins are basically RNA helicase. Now it uses energy to move along the RNA molecule and catch up with the RNA polymerase. Then it leads to the dissociation from the DNA template. Right? Right. So the next set. Question 13. Translation occurs on which cellular structure? Oh, it's very direct. It's the ribosome, no doubt about it. So option C. Question 4. Which RNA carries amino acids to the ribosome? Which RNA carries amino acids to the ribosome? Yes, the tRNA, that's the transfer RNA molecules, are responsible for carrying the amino acids to the RNA. So first there is a tRNA that has an anticodon that matches with the specific codon on the mRNA, and ensuring that the correct sequence of the amino acids are put up in the growing polypeptide chain. Fine. The question 15, so for question 14 it is option C. Question 15, the Shine-Delgarno sequence is involved in. The Shine-Delgarno sequence is option termination, initiation, mRNA processing, protein folding. So we know that there has to be a starting point. So the Shine-Delgado sequence is the ribosomal binding site in the prokaryotic mRNA. It recruits the ribosome to the mRNA to initiate the protein synthesis. So the initiation of translation starts from the Shine-Delgado sequence. So that's option B. Now, which ribosomal site holds the growing polypeptide chain? Which ribosomal site holds the growing polypeptide chain? A, P, E, T. So we talked about it. The A site is the amino acid site where it identifies the tRNA. It goes and sits over there. So the charged tRNA enters from the A site. The growing polypeptide chain is there in the P site, where after the methionine, after the F-met, there is the addition or the elongation of the amino acids after it has entered the A site, and finally through the E site the mature polypeptide chain, after it has reached the stop codon and it has found no more tRNAs for against it, it stops over there and exits to the E site. So the elongation chain is the P site or the peptidyl site. A is the amino acid site, P is the peptidyl site, and E is the exit site. And basically the last set of questions. Question 17. Peptidyl transferase activities associated with small ribosomal subunit, large ribosomal subunit, tRNA, mRNA. So what is the peptidyl transferase? So peptidyl transferase is a ribozyme. That's an RNA enzyme that catalyzes the formation of the peptide bonds between the adjacent amino acids during translation. And this activity is located in the large ribosomal subunit, that is the 50S subunit in prokaryotes, and in eukaryotes what do we call it? The 60S subunit. So option B, that is the large ribosomal subunit. Question 18 is, which factor is responsible for translocation during elongation? EFTU, EFG, IF and RF1. So what is the role of EFTU? EFTU is the elongation factor thermo-unstable. It is responsible for delivering the amino acid tRNA to the ribosomal A site during the elongation phase of translation, ensuring that it's correctly decoded. Then is the EFG. EFG is the elongation factor G, which catalyzes the translocation step where the ribosome moves along the mRNA by one codon and subsequently acts in the ribosome recycling to disassemble the post-termination complex. IF3 is the initiation factor 3 that binds to the 30S ribosomal subunit to prevent the premature association with the 50S subunit of ribosome, and it helps in mRNA binding checking the fidelity of the initiated tRNA during the initiation process. And RF1 is the release factor 1. It's an A class one in release factor that recognizes the stop codons, the UAA, UAG in the ribosomal A site, and promotes the hydrolysis and release of the completed polypeptide chain from the tRNA. So which factor is responsible for translocation? That is option B, that is EFG, or elongation factor G. Now finally, termination of translocation is signaled by. So termination is signaled by, we know all of the that it's the stop codon, because there are no tRNAs against the stop codons. So there are no tRNAs for the stop codon. So when it does not encounter a tRNA at the A site, then there are release factors that throws it out from the E site of the ribosome. The matured polypeptide chain is out. Right, so stop codon. Which molecule directly determines the amino acid sequence of a protein? DNA, tRNA, mRNA, or rRNA? So which molecule determines the amino acid sequence of a protein? It's not DNA, because DNA is the basic unit of the genetic information. It acts as a blueprint rather than a direct template for the ribosome. So it must be converted into mRNA before the protein can be made. tRNA, definitely not, because it acts as an adapter. It carries specific amino acids to the ribosome, does not determine the sequence. It just matches its anticodon to the instructions already present in the mRNA. Then you have mRNA, which is the correct one over here. So based on the template sequence of DNA, the mRNA is produced, and the ribosome uses this to allow the tRNAs to come and elongate the polypeptide chain. And rRNA, it's a ribosomal RNA, so definitely not. Right? So option C, which is correct. So that's all. If you have any doubts, you can ask me. So it's a recorded lecture, and it will be uploaded in YouTube. Anytime you're having a doubt, you can go back and check from the YouTube channel, the YouTube link that will be shared by the NPTEL team with you, and I will be uploading the presentation as well, so won't be that of a problem. Fine. Okay then. Thank you. We will again meet in the next week.
Thank you, sir.